Okay, hi. This is the second half of chapter 10. We're going to finish that lecture. So we're starting with this learning outcome here. Describe the impact of injury on endothelial and epithelial cells in various organs. So we're basically, talking about cellular injury in this chapter. Okay, so if we talk about epithelia, right, these are cells that will line cavities, right, and cover flat surfaces, like epithelial cells right in your skin. They're able to differentiate and undergo rapid proliferation to replace injured cells. So this is something that's happening all the time, right? They have this ability, if they're damaged, if a cell is damaged, the body is able to replace these cells, right? An ongoing process. If you're talking about things entering or leaving the body, like through your GI tract, they have to go through those epithelial cells, right? Not in between, because otherwise you have problems. We talked about that before, these tight cell junctions. And so super important that things are going through these cells. If we talk about endothelial cells, these are cells that line blood vessels. It says they're gatekeepers to the tissue compartment. Obviously, right? They're lining the blood vessels, and they're the cells that are in between what's in the blood and what's in the tissue, right? Again, things need to get from the blood to the tissue. They're going to go through those endothelial cells. If you have impairment of those endothelial cells for whatever reason, it can lead to edema, hypertension, right, which is increased blood pressure, abnormal vasoconstriction, because again, what's in the blood is not, able to leave the blood or what have you and get into the tissues. It says initial step in atherogenesis, right, which essentially is the formation of these clots in blood vessels, right, and can lead to heart disease and stroke. Again, if you have impairment of these endothelial cells, and then you have accumulation of lipids and things there, that's the sort of first step there in terms of forming a clot, right? The clot isn't just the lipids, but it's also the endothelial cells there that maybe start secreting inflammatory molecules and things like that, okay? And so that's how those are involved in that process. If we talk about injury specifically to epithelial cells, right, can happen for various reasons. So insect bites, scrapes, cuts, and blood may leak out, and then microorganisms may actually be able to enter. And so results of the injury, and again, this is the body's response to repair that injury, right? You'll have release of factors that will activate fibroblasts. They'll produce collagen, fibronectin, all in an attempt to form a plug, right? So that blood doesn't keep leaking out. And so these epithelial cells, a lot of them have this ability to be continually recycled, okay? And so if you talk about the GI, the respiratory tract, you're going to have this continuous recycling if there's an injury, and the body has the ability to replace these epithelial cells. However, if you have inappropriate stimulation of these epithelial cells to divide and to produce more, that's how we can lead, how it can lead to tumors. Again, they have the ability to divide and make more cells because we need that ability to repair, these lost cells due to injury, right? However, if it becomes too much, it can lead to tumor development. Okay, so if you look at this figure here, this is showing you epithelial injury and repair in airway disease, right? So you can see here, right, this sort of epithelial cell layer here, right? And here's some injury. And again, you get a loss of cells here, right? And so what's going to happen is you're going to end up replacing those cells, right? So you can see there's more fibroblasts that end up proliferating here, this sort of increase in these basal cells. And then ultimately, you're going to get those epithelial cells to replace the lost epithelial cells there. Okay. So let's talk a little bit more about cellular responses to injury, okay, and cellular repair processes, okay? So this is all talking about morphological changes in the cell, okay? So cell morphological changes that happen in response to some sort of cellular injury, okay? So this says response to reversible injury. Remember, we talked about reversible versus irreversible injury. These changes that occur to the cell are going to allow that cell to recover and to be able to function properly, okay? So one thing is the size can be altered, right? So if you talk about atrophy, that's a decrease in cell size, okay? So if there's a decrease in work demands or there's some adverse environmental conditions, it is advantageous for that cell to decrease in size, because if it decreases in size, then there's not as much, energy need, right? And also just need for resources, right? So it allows that cell to be able to survive in that whatever environment, okay? If you talk about, atrophy in muscle cells, right? This can happen due to disuse, right? Or if you're paralyzed and you can't use that, you can't use an arm, you're gonna have atrophy, okay? So again, that's due to these cells decreasing in size. If you talk about hypertrophy, this is an increase in cell size, and so this will happen because of increased workload, right? Specifically, if you think about cardiac and muscle tissue, skeletal muscle tissue, you'll see this. So it can be a normal physiological response from working out, right? You lift a lot of weights, obviously your muscles get bigger. That's because those cells increase in size to, again, meet those physiological needs. However, it can be pathological as well. So a result of disease conditions. So for example, you can get this thickening of the urinary bladder due to obstruction of urinary outflow, right? So if there's some obstruction there, it then causes an increase in cell size in the urinary bladder, which leads to this thickening, okay? You can also see it happen with, as a compensatory mechanism, right? So if you lost one kidney, the other kidney will actually increase in size, or those kidney cells, right? Those cells there will increase in size to compensate for the loss of the other kidney, okay? Again, these are all changes in cell size. Also, you can have changes in cell number, right? So if you talk about hyperplasia, that is an increase in the number of cells in a given tissue, right? This is something normal that happens during pregnancy, right? An increase in the number of cells. It's not just in the size of the cells, it's actually in the number of the cells. If you talk about sort of form, right? So something called metaplasia is that one cell type will be replaced by another cell type. So hyperplasia, it's just an increase in those number of cells, the same cells that were there before. In metaplasia, you're going to get another cell type replace what would be there. So if you even think about diseases like muscular dystrophy, right? There's a dysfunction of the muscle cells, and what ends up happening is you get other cell types like epithelial cells that will replace those lost muscle cells. However, those don't function as muscle tissue, right? And so you can see the problem there. So this does happen in a lot of different scenarios. Dysplasia there is deranged cell growth of the specific tissue. So you get this weird kind of disorganized cell growth. Cells will vary in shape and size, and this is believed to be a precursor of cancer, right? So if you looked at you went and you got a biopsy or something like that to look at that specific tissue, if a clinician saw this sort of deranged cell growth, it would indicate that perhaps this could end up being, cancerous. These cells can be. So it's sort of an indicator. Autophagy is a term that sort of describes a process where a cell will break down its own contents to be able to survive in times of stress. So again, it's a sort of a compensatory mechanism, and I don't know that it's really a change in form, so maybe it needs to kind of be a separate bullet here. But basically, the cell needs to be able to do it. However, if there's too much autophagy going on, it can lead to cell death, right? And sometimes that's where it should lead, right? So it's tightly regulated. The cell, again, starts breaking down maybe some of its cellular organelles to be able to generate enough energy to function. Like I said, it can lead to cell death. And again, it's sort of linked to cell death pathways, okay? And so there are a lot of studies out kind of recently that sort of look at autophagy and regulating autophagy as a potential therapeutic target for various types of diseases. Okay, so if you look at these morphological alterations, we've talked about these, right? Here are some examples of that, right? Normal, hyperplasia, increase in cell number; atrophy, decreased cell size; hypertrophy, increase in cell size, right? Metaplasia here, conversion of one cell type to another, right? So these cells are lost, but what's replacing those lost cells is not the correct cell type, or it's a different cell type. Dysplasia here, you can see this disorderly sort of growth. And again, as this is a precursor to indicating that this tissue then could become, cancer. This could become a tumor. Okay, sorry. So let's continue with this. So this is intracellular accumulation. So again, if there's some sort of cellular injury, right, it can lead to a buildup of substances that the cell can't immediately use or can't get rid of, right? So you can lead to this buildup of stuff in the cell. So for instance, water is one, right? Obviously we know there's lots of water inside and outside of the cell. It needs to be able, it's transported across that plasma membrane all the time. However, it could end up being built up inside the cell. So it says hydropic degeneration. So due to ischemia and to other chemical agents, you can get this buildup of water. Buildup of lipids says most often occurs in the liver, right? As a result of alcohol intake, obesity, starvation, you'll get this buildup of lipids inside the cell. Again, the cell should be breaking these down and using these to generate energy, but for whatever, there's this sort of breakdown in metabolism or this sort of problem with the metabolism that you're getting this buildup there. Calcium buildup, right? You see this in dead or dying tissues. You have this sort of calcification of those tissues that creates this sort of hard, you've heard of like calcium deposits and things like that, this kind of hard kind of part of the tissue. Glycogen, you can get a buildup of glycogen. So in diabetes and glycogen storage diseases, again, glycogen in the liver it's a storage molecule for glucose, right? So when the body's blood glucose levels are low, the body wants to break down that glycogen to then form glucose, right? So that glucose is available to use. And so in diseases where you have a problem with glucose metabolism, you'll see a buildup of glycogen. There's also something called hyaline, which is this sort of protein-containing debris that can build up in the cell. Pigments like melanin can build up in the cell as well. Again, these all indicate some problem, right? You shouldn't have buildups of these things inside the cell. Iron, stored in the liver and heart cells, can also build up and then can lead to heart failure or fibrosis, again, which is kind of this hardening kind of the tissue, that fibrosis, okay? If we talk about cellular responses to injury, what can some of these subcellular changes? So it says alterations in the number, the size, the shape, and the function of organelles like the smooth ER. Again, the smooth ER is involved in lipid synthesis, right? Mitochondria. We know the mitochondria is involved in forming ATP, okay? And so, again, if there's alterations in metabolism or some injury to the cell, there's going to be changes in these types of organelles. Cytoskeleton, remember we said is important in maintaining cell shape, but also in sort of forming junctions between cells and what have you. It says responses there, so acute lethal injury, chronic injury, adaptive response. You can have all these different responses by the cell to injury, okay? And so some of those responses can be, you can have inflammation. We already talked kind of about that. And so if you have increased inflammation, free radicals, we already talked about free radical formation can enter damaged tissues. You can then have necrosis occur. Okay, and basically, you know, it's cell death and necrosis, death of the tissue. Okay, in terms of cellular repair processes, you know, if there's an injury, the cell is going to initiate these repair processes. They don't always end up working, and if they don't, then the cell may end up going on to die. So it says clearing out damaged proteins, right? So if there's something happening to the cell where you have a bunch of damaged proteins, the cell does have the ability to get rid of those damaged proteins. If the cell doesn't get rid of those damaged proteins, they're going to obviously not function properly. They also can induce other proteins to coagulate and form and cause them to be dysfunctional. So it's super important that the cell gets rid of them. It can then get rid of those damaged proteins and then produce new functional molecules, right? Again, if the cell can't do any of this, it will have to go on to die. And so that's what the whole point of this is, to explain that this is a normal process that does occur after injury, but that doesn't mean that it always is successful. Okay. So now, so like I just said, right, if these processes aren't enough, right, we then, the cell may have to go on to die. And so you can kind of think of two types of cell death here, right? So there's something called necrosis and also apoptosis, okay? So we're going to just talk a little bit about why those two are different or how they're different. Okay, so necrosis is cell death in part of a living organism. It's unregulated. That's the big difference. So it's unregulated, as I was saying, necrosis, just as enzymatic digestion of the cell components, loss of cell membrane integrity. So if you look at it under the microscope, you'll see this sort of breakdown of the cell membrane. That's indicative of a necrotic cell. Initiation of inflammatory response, so you'll see all these inflammatory molecules as well. It says gangrene, right? We've all heard of gangrene. We see this in diabetes and basically it's a mass of necrotic tissue. Okay, if you talk about apoptosis, this is programmed cell death. So this is kind of a normal process that has to happen in the body to get rid of these cells that are dysfunctional, right? So you can think of the necrosis as kind of these cells are dying because of pathological kind of processes in the cell, right? Like we kind of talked about there, forms gangrene, right, where you have this sort of dead tissue that forms. In apoptosis, this is tightly regulated and programmed. Okay, so that's why it's saying here it's highly selective. There are two different distinct pathways. An extrinsic pathway where you have specific receptors that become activated based on molecules that might bind to them in that extracellular environment. Like this is giving an example of TNF here, tumor necrosis factor. There's also an intrinsic pathway where that means inside the cell, right? If something's going wrong, and again, it's something that the cell can't repair, things like DNA damage. DNA damage, again, can normally be repaired by the cell, but sometimes the DNA damage is too much for the cell to repair. Sometimes there's too many reactive oxygen species that form, okay? Sometimes the decrease in ATP levels is too much, and so it is in the best interest of the cell to go through apoptosis and go through this programmed cell death. Okay, so extrinsic is something on the outside of the cell triggering the apoptosis. Intrinsic is something from the inside of the cell that's triggering that apoptosis. And so if you look at a picture of this, this basically kind of is showing you necrosis versus apoptosis. So like I said, you can look under the microscope and there's certain differences that you can detect to tell whether it's necrosis versus apoptosis. There are also other types of tests or markers that you can use to distinguish whether a cell is going through apoptosis versus necrosis. So again, you can kind of see here some of the morphological changes that occur in a cell that's going through necrosis. And you can see the presence of the inflammatory markers, or these are just inflammatory cells, right? White blood cells around. Here you don't see that, right? And so this you kind of see the cell fragment, okay? It says chromosome condenses because the cell knows that it's going through this cell death. This is intentional, right? You get these membrane blebs. Like I said, these are things that you can see under the microscope. And again, the fragmentation of the cell. And then these apoptotic cells in the fragments get engulfed by other, white blood cells that are phagocytic cells. Okay. Now we're going to talk about the sort of normal lifespan of the cell and kind of just some things about the sort of molecular basis of aging, meaning what things are changing, what types of molecules and things inside the cell that change as a result of aging, okay? So if we talk about normal lifespan in the cell, it's going to definitely vary depending on cell type, where the cell's located, and what have you. So it could just be hours, right? It could be weeks, it could be months, it could be years, again, depending on what cell type you're talking about. When you talk about neurons, right? Those neurons that you're born with are the ones that you have until you die, okay? And so that's an example of a cell type that has really long lifespan, okay? And so if we talk about the molecular basis of aging, again, we're talking about molecular, we're talking about molecules, right? So it says focuses on mutations in gene expression. So things that are, it could be various things, right? So you could have mutations that occur in the DNA that then lead to sort of non-functional or misfunctional molecules that are formed, right? It says replicative senescence. I think we'll talk about that in a second. Basically, the cell only has the ability to divide so many times, okay? So once it sort of reaches that limit, that's sort of believed to be involved in aging, right? And why you start to see a decrease in function in organs and organ systems as you get older. Telomeres are regions at the end of the DNA that basically protect the DNA. So again, when a cell divides, it has to replicate its DNA. The ends of the DNA don't really have the ability to be replicated, right, to the extreme ends. And so at the extreme ends of the DNA, there's sort of sequence that's not really coding sequence. And those are these telomeres. And over time, they get shorter and shorter. And so what happens when the DNA is replicated and these telomeres are really short, there ends up being some parts of the DNA then at that point that are needed that don't get replicated, okay? And so then you're missing, molecules that you need. And so again, leads to dysfunction. Free radicals, again, we talked about free radicals before. And so if you keep accumulating them over time, can lead to cell death, which then again can lead to tissue misfunction and organ misfunction. We also talked about apoptosis. Again, you can see if there's an increase in apoptosis overall, kind of when it shouldn't be, and you have loss of cells, it's going to lead to aging, right? Again, some kind of more molecular things that can happen or that are believed to be involved in sort of aging, impaired protein homeostasis. Again, you're not making enough proteins to sustain what needs to happen metabolically in the cell. Defective nutrient sensing. So even though the nutrients are outside of the cell and they're available, the cell's not able to, they're not able to bind to the receptors, the cell's not able to sense them and not able to use them. An accumulation of damaged DNA, same idea. The cell's not able to repair that damaged DNA, leads to problems. Right? And I kind of said these kind of along the way. If you have cells dying in a certain tissue, in a certain organ, you're going to end up with organ dysfunction, organ system dysfunction. Okay? And if you talk about just cell function, kind of, specifically that some of these molecular changes lead to, defects in oxidative phosphorylation by the mitochondria. Again, it's going to severely limit the amount of ATP that can be formed. Synthesis of nucleic acids and transcription factors. Again, that's going to greatly affect gene expression. Okay. Again, cell receptors, cell proteins. We talked about these things along the way. If you talk about diabetes, something that kind of happens over time. A lot of times it's due to a dysfunction of the receptor, right? So you can either, if you're talking about misfunction of a certain process, like glucose metabolism, it could be due to the fact that the protein isn't made, something like insulin you don't have enough of, or it can be the fact that that insulin receptor, either you're not making enough or they're just dysfunctional in some way. So you got to think about both levels here, both the receptor and the protein or the molecule that binds to the receptor. Okay, and so that finishes chapter 10. Also, remember to kind of look in the notes because there are sometimes some definitions in the notes part of the PowerPoint. Okay, thank you.